Hello everyone, this is the teaching video to help you learn about making observations under microscopes. So you have learned the practical aspect of this. When you go to the lab, you know how to use the microscope and you know how to make well-focused low power observation and high power observation of living things, and you also know how to make specimens onto a glass slide. So these practical aspects would be asked in the test as well. So I hope you enjoyed the lab session and I hope you learned from what you have done. Remember to go to this video to revise the steps in using light microscopes, and also read the textbook pages about the design of the microscope as well as the procedures involved in using a microscope. Well, the words here list out what we expect you to know about light microscopes. I'm not going to read through every sentence because every question in this worksheet is going to correspond to these learning objectives. So let us go through the questions in the results and discussion part. And remember, you can follow this video to complete your worksheet, but remember, don't just copy the answer. More importantly, you should understand why or how we come up with answers like this and some important principles behind. All right, let's start. The first thing that we want you to learn about microscope is the property of the image you can see. Now remember that when you are observing under microscope, the light passes through the specimen or sample from the bottom, and then it passes through two lenses, the objective and the eyepiece, and then eventually reach your eyes. So these lines represent the path of the light going through. There are two important properties of the image as a result. The first property is the orientation or 嗰個位置感 of the image. If the slide has a letter F like this, I think all of you remember how the letter F should look like. Can you pause at the moment and think about the answer? You should realize that the letter F would be inverted in both directions. It's upside down, and in terms of left and right, it's also inverted as well. So we describe that the image formed by light microscope is inverted. Comparing with the original object. Now, this also has an important implication. It means that when you want to move the image to one side, let's say if you want the image to go to the right, you actually have to move the position of the slide in an opposite direction because the image is inverted. Now the second property of the image is that the image is magnified. But by how many times the image is magnified, it's important to know, because eventually we want to know the actual size of the specimen or the sample. So that's why we need to know the exact magnification power during observations. Now, there are two lenses, remember, the eyepiece and the objective. Each lens would magnify the object by a certain times. In the first case, and low power observation in our lab, the eyepiece magnified the image by 10 times, while the objective magnify the image or the object by 4 times. So eventually the image that you can see would be magnified by 40 times, because this is a multiplication. Now similarly, I think everyone now can work out the high power situation as well. So I'm not going to write the answer, but let you think. Now talking about magnification, let us jump to the next page first, and I would like to at once show you how magnification is important in our calculations of the actual size. Now, in photomicrographs like this, we would present the magnification in two different ways. So either we will write it like this in the title or stated within the photo, or we would use a scale bar like this to tell you the magnification. So let me explain a little bit more about the scale bar. The scale bar here means that this certain length in this photo represents 100 UM. UM means micrometer. It is a unit that is much smaller than cm or mm. So these are the common units of length you use in daily life, but since cells are very small, so we use a much smaller unit for the length. So let's say this question asks you to calculate the actual length of cell X. I hope you have already tried and think about how it works. So here are the answers coming up. The first thing that you need to do is to measure the length of the cell Y in the picture. So this is the measured length here. But we set it aside at the moment, all right? We will use it later on. Another thing that you need to work on is the scale bar. I need to first work out the measured length here. So let me say this is XCM for cell X, and this is, let's say, YCM. So before we really set up the calculation, let me explain a little bit in Cantonese, because this would be quite difficult for many of you. 我哋嘅思維方法係咁嘅，如果每 YCM 就代表100 micrometer in the picture。咁你可以幻想下，如果我有2倍嘅長度，我有2Y 咁長嘅話，個 YCM as measured length 到頭來就係代表100個 UM in actual length 的話，咁你可以幻想下，咁如果喺個圖畫我度到個 length 任何一個地方嚇，我度到個 length 係2Y/cm 的話，咁請問 actual length 係幾多呢？我相信呢個唔難啦，係咪啊？咁就變咗200 UM 嘅咯喎。好啦，你可以幻想我呢個故事可以去到3Y、4Y、5Y、6Y 係咪？咁如果有一個數而家我度出嚟係 XCM，咁到底藍色呢個數字應該係幾多呢？我俾你諗一陣好嗎？係，所以我哋首先要諗下，到底 x 係 y 嘅幾多倍咧？嗱，喺頭先嘅例子你會見到嘅，如果度出嚟個 measured length 係 y 嘅2倍的話，咁我只要將呢個2倍呢個數乘100，咁啊可以得出個真正 actual length 係200咯。又例如我再講，例如如果呢度係5Y/cm 的話，咁相信大家冇難度嘅，因為你知道只要將100乘5，係嘛？答案就係因為佢係呢個 scale bar 裡面，佢係 Y/cm 嘅5倍，所以個 actual length 就係100 micrometer 嘅5倍。好啦，當我哋要計呢個問號嘅時候咧，亦即係計緊個 actual length of cell x。咁嘅時候應該點計算呢？咁我哋首先要知道 x 其實係 y 嘅幾多倍。咁即係應該加減乘除咩數呢？我俾5秒你諗下。我要知道 x 係 y 嘅幾多倍，應該係加減乘除數，邊一種呢？你應該知道係除數啦，第一。咁邊個除邊個呢？諗清楚咯喎。x over y 啊，定係 y over x 啊？我亦都停一陣俾你諗下，停5秒、10秒俾你諗下。俾個提示你啊，咁你頭先計呢個5係 5y 除 y 啊，即係5y over y 啊，定係 y over 5y 咧？嗱，諗清楚下。好，相信你已經得出嘅結論咧，就係 x over y。呢個咧就係代表而家我 measured 嘅 length 比起呢個 scale bar 裡面嘅 measured length 大咗幾多倍？大2倍，咁呢個數咪乘2咯；大3倍，呢個數咪乘3咯。大咁多倍嘅時候，只要我將呢個數字乘返100 UM，咁我就會得出嗰個答案嘅。OK， 實際數字我希望由你自己去做一次。OK，so I hope this calculation illustrates the general rules in working on magnification. So calculate the ratio between the measured length of the scale bar and the measured length in the picture, and then multiply it by the actual length shown in the scale bar, and you'll get the actual length of the cell. So this is one of the ways to find out the actual length of anything that you see under light microscope. But there is another way. Sometimes the picture doesn't show you the scale bar, but only give you the magnification power. It is actually possible to use this to find out the answer or find out the actual length, and the details of the calculation is actually shown in textbook page 7. So you are required to go and read this page and learn how to calculate. In case you don't know about, I'm quite sure you can get the answers easily, but in case you are not sure, please go ask your teacher for the exact answer. So that's all about the properties of the image. Again, we conclude here first. The image that you see under light microscope is inverted and also magnified. And the exact magnification value would allow scientists to calculate the actual length of living things or any samples you see. And that is important. Okay? Now let's come to learn how to present our results when using light microscopes. So we have two types of observations, either low power or high power. Can you tell which picture is low power and which picture is high power? And the reason behind, why do you think a certain picture is low power or high power? Can you try to explain? I would say this is a low power photomicrograph. Even I don't see the questions. Even the question doesn't tell me. I know that this is a low power because many more cells can be observed, but with less details. Okay, and this is the characteristics of low power observation. Low power observation is, let me explain high power as well first. So this would be high power observation because you can see less cells, but they are in a better details. So you can see the nucleus very, very clearly, and you can even see some solid organelles, circular organelles within the cell, and you can never see in this picture, right? So which power of observation is better? I give you 5 seconds to think about it. Which type of observation is better? Well, many of you may think that, all right, high power is better because you can see the cells more clearly, you can see a lot of details. Well, that's true. But I won't say low power is bad. Actually, low power is also good. Now, both these type of observations would be good, depending on the situation and your purpose. If you want to see the overall design of the epidermis, in this case, how the cells are arranged, how the cells pack themselves, or even you want to know how many types of cells are there in a certain specimen, a low power observation would be better for these situations because you can see many more cells there. So remember that both types of observation is good depending on the situation. This is good for looking at the overall pattern, and this is good for high power observation is good for looking at details Now, when we present photomicrographs like this, we always, or even drawings, we need to give titles to the pictures. The title should include the name of specimen and also the total magnification, because you need to tell me how magnified it is. Otherwise, I can't imagine the actual thing, how it would look like and how large it would be. So here, the title for this photograph would be, I'm observing red onion epidermis. And I need to address the magnification. So I go back to part 2, question 2, and see how many times the object is magnified. So it's 40x. So I write it here. Now, please also try to complete this part. These 3 boxes ask you to label these 3 organelles. So, and the jelly-like substance here, and also the thick line you see here. So I'm confident that all of you can come up with the correct answer. Now please allow me to skip to the last page of the worksheet, as we have been talking about low power and high power observations. Can you fill in this table based on what we have discussed? So low power observation allow us to see a larger area of the specimen, but with less details. While high power observation focus on a smaller area of the specimen. So you can see that this circle, the view here, only covers several cells. So it's actually maybe zooming in into this area to get this high power. So I hope this can help you to understand that high power covers a smaller area of the specimen. But since the magnification power is higher, there are more details to be seen. So last thing that you need to realize is that you can see from this photo that the high power magnification is much dimmer while the low power observation gives you a brighter image. So we got to explain why it is. And I think this picture would help. Again, remember that high power observation is just observing a smaller area. So what? If you look at this photo again, about how light microscope works, you will realize that light is passing through the bottom, from the bottom of the specimen up. So you can imagine that when you can see this circle, it means that all these areas allow light to pass through. So in low power situation, it means that a greater area allows light to pass through. in this case, you can expect the image to be brighter. However, under high power observation, since the area is much smaller, then these two light rays actually doesn't go to your eyes. Only the light in this area would go to your eyes. So therefore, it means that less light is collected and enters your eyes. So that's why high power observation is usually dimmer. How can you solve this problem? You can solve this problem by turning the lamp under the microscope to become more powerful, so that less light rays come through, but let's make each light ray much stronger with a higher intensity. Remember that there is a tab at the bottom of the microscope that allows you to make the light stronger. So that's why every time we switch from a low power observation to a high power observation, whenever we do this, we need to turn the light to become stronger intensity, so that the image of the high power observation would be bright enough for us to see clearly. So with these features in mind, we can now see that when I'm searching for a particular thing to see, when I'm searching, it's better to look at a larger area of specimen. While if I'm looking for more details, it's better to use high power observation, simply because the magnification is higher, so that it can give you more details. While a larger area allows easy search. So remember to write the correct answers in full sentences when you submit your work. Well, so far we should have covered all the aspects that we want you to learn about microscopes, properties of image, how to present your results, and knowing that you should use low or high power observation under different situations and the reasons behind. The last thing that we want to teach is for this particular session, in that we observe red onion cells. Now, when you look at a red onion, the whole surface is red. Agree? So when you see the epidermis like this, you should find it strange, because you would see that a lot of areas here are not red. They are actually colorless, with the cell walls remaining. So what happens to these cells? Are they naturally like this? Well, I think you will know that something have happened to these during the preparation of the slide. They are not naturally colorless, but something happened when we work on it. The red color comes from the cytoplasm that contains red pigments. They stay in the cell because they are surrounded by a layer of cell membrane. Now, it's not about cell wall, because if you remember, cell wall is fully permeable. So therefore, even the cell wall is here, but if the cell membrane is damaged, you can imagine everything in the cell can leak out through the cell walls. So therefore, the cell Y here and all the colorless cells here, they are not naturally colorless. You can imagine that all the cytoplasms that contains the red pigment leaks out. But what caused this? I've told you is because something has been damaged. Is it cell wall or cell membrane? Think about what we have discussed just now. But I want to trace to an earlier reason. Why are they damaged? I think this picture would tell you the answer. When you peel the epidermis, you may have physically damaged some of the cells when you tear the epidermis off. So that's why I would say this is the first reason that caused all these problems. Now, don't think that you have done anything wrong. It's not your fault, because when we have to tear it, this always happens. Even teacher like us, when we tear it, it happened. So it's just make sure you understand why that happened. It's all right. Okay? Well, some of the students in our class actually made some strange observations like this. I would say these photos shows very, very good observations, while these look strange. So what happened? It's all the problems in preparing the slides, caused by problems in slide preparation. These and these are actually, they are not vacuoles, but they are actually gas bubbles. They are not naturally present in the epidermis, but rather, if you remember, there is a step that when you have a glass slide and you have a piece of red onion epidermis here, you actually put it on a drop of water. Do you remember? And then you cover it up with the cover slide. You do this, if you remember, you do this by lowering the cover slide slowly. So when you let the cover slide fall down to cover, accidentally some gas bubbles may be trapped. So that's why you can see these gas bubbles are of random sizes, because they are accidentally formed. So how can you avoid this trouble? you can avoid this trouble by lowering the slides very slowly so that you allow the gases to go away before the slide is covered, and not to trap any air inside the water. So this is a very common situation when preparing the slide. Well, another problem is like this. So what do you observe? What are these ugly lines here? What are these ugly lines? Now you can see the area that we want to observe is here. It should look very pretty like these areas. But now it seems that you are blurred by all these lines. And if you see carefully, these lines seems look like cells or cell wall. Agree? Some of them are colorless, some of them are slightly red. So what's the problem? Now the problem is that, can you guess? The problem is that the epidermis, I would say, what you see here are actually cells. But they are not very clear because they are not at the same layer as this. Your light microscope will only focus on a certain plane, 某一個平面啦嚇。So if the red onion epidermis is very thin, containing only one layer of cells, you'll see beautiful pictures like this. This is only one single layer of cells. But now the problem is that if you accidentally, or if you don't peel the epidermis very nicely, or when you put the epidermis into the water, if you let it fold up, and press onto, so let me say this is the folded area, and let it cover up. Now you can see that there is an extra layer of cells covering up the layer that you want to see. So this is the extra layer, while this is the layer that you want to see. So the red area here may be the layer that you want to see, while at the same time it is covered up by extra layers because of the foldings. Those layers are made up of cells as well, agree? So that's why you see all these ugly lines here. They are all extra layers of cells covering up the layer that you want to see. And the reason that there is an extra layer, it's because the epidermis is folded. So how to prevent this problem? So that's why when you before you put the cover slide on, you have to use forceps to unfold these layers so that there is only a single layer present in the slide. So this completes our discussion. I hope you enjoyed the session. If you have any questions for these answers, or if you don't understand, please feel free to contact your teacher anytime soon. Okay? I hope everyone can, all these things we teach in this video will be covered in our first uniform test. So good luck to you all. Thank you.